Great. I think we're going to go ahead and get started. So thank you so much, everyone, for joining. My name is Hannah Schuyler, and welcome to today's webinar, Fast, Meaningful Insights, Simplified Omics Analysis for Biologists. As I mentioned, my name is Hannah Schuyler. I'm a Director of Business Development here at Almaden Genomics, and joining me today is Kit Furman, who leads our product and development team. And joining us from the BigOmics team, we have Axel Martinelli, who is their head of biology. So thank you again for joining. A couple things before we get started. This webinar is being recorded and will be sent out to all attendees after and will be available online as well. Please enter questions that you have for the presenters in the chat box, and they will be addressed at the end during the live Q&A session. So in terms of what we'll be discussing today, first we'll be highlighting our end-to-end solution and the importance of being able to process your raw sequencing files all the way to tertiary analysis with both of our solutions. Then Kit will give an overview of the data set we'll be analyzing today, which is a multiple myeloma data set. Then we will go through a demo of how you can actually use the Genome and Omics Playground softwares to process your data. And then lastly, we'll leave some time for Q&A. So if you have questions, again, please enter those in the chat box for us. So when it comes to analyzing data, there's many complexities that make it very challenging. There's, of course, massive amounts of data across different teams and work streams, making this very difficult to manage the data in one secure place. There's also often a communication gap between both the biologist and the bioinformatician. I'm sure as many of you know, it's a very iterative process when it comes to analyzing the data. The biologist often relies on the bioinformatician to analyze the data, so they're not necessarily self-sufficient. And specifically for tertiary analysis, there's a surplus of open source solutions that aren't always updated, regulated, or organized, making it very, very difficult for the biologists to do the routine analysis themselves. This, of course, slows down the data analysis process and time to insight and discovery. And then lastly, there's a need for transparency and organizational systems to track and reproduce methods for experiments in the future. So with all of that being said, we're looking to provide an end-to-end solution for researchers. Of course, that first step really begins with data, right? So it's whether you have your in-house sequencing data or public data sets that you're looking to analyze, all of that data can be brought in to the software, and we provide the ability for users to securely manage the data through different workspaces. The next step is really around data processing. So you can start with some of our pre-built workflows and use as is. You can modify it, or you can build a pipeline from scratch. The next step is focused on the tertiary analysis, and this is really where our partners BigOmics come into play and excel at this type of analysis. So they have access to over 100 different visualizations that allow you to compare data sets. You can perform pathway analysis, gene set enrichment analysis, explore the drug connectivity and mechanism of action, and many more types of reports. And then lastly is the service component. So we're really focused on providing kind of that white glove treatment in terms of customer support and training with all of our customers. We also can build custom bioinformatics workflows and curate and clean data sets for processing. So today we're going to talk through how you can actually use Genome for data processing and automation, and then take those outputs from the software, which are process data file, and then visualize that data on the Omics Playground. So before I pass it over to Kit to walk through the data set, I'd like to poll the audience and get a sense of where you guys are at with RNA-seq data. So in just a second, you should see a poll come up. So when do you anticipate you will be analyzing RNA-seq data? Right now, within the next 3 months, within the next 6 months, within the next year, or I don't need to process any data, I'm here to learn. Looks like we're getting some answers coming through. It's kind of all over the place. It looks like majority of you so far, before I end the poll, have data, I would say right now, within the next 3 months and within the next 6 months. So it looks like a lot of you have data, which is great to hear. So yeah. Thank you everyone for participating. And with that, I am going to pass it over to Kit to walk through the multiple myeloma data set that we'll be talking through today.
Thank you, Anna. Again, my name's Kit Furman. I'm the head of product and development here at Almaden Genomics. Excited to share with you the Genome platform. Today, we're going to use this study on multiple myeloma to walk through genome and BigOmics. This is a good publication that explored the ability of with afferent A to treat resistant and sensitive cell lines that are resistant or sensitive to glucocorticoids. If you don't know, multiple myeloma is the second most common blood cancer. And a common problem is that patients will become resistant to whatever treatment they're on, forcing doctors to change treatment. And this process, can go on for more than 10 years. So it's a very treatable disease, but it does force doctors to just go through repetitive lines of treatment, whether that's immunotherapies, glucocorticoids. And so doctors and scientists are constantly on the lookout for new treatments. And so with afferent A is a small molecule that targets tyrosine kinases. And the hope is that this can be another treatment for patients and offer them a stronger response against the tumor. So next slide, please. So we're going to use this data set as an example to walk through the genome platform. We're going to download the study data from Geo. So Genome allows you to pull in data from public sources. We're going to run the RNA sequencing guided workflow. And then we're going to review the sequencing results and then, importantly, download those counts for BigOmics analysis. And so preparing for RNA analysis, I just want to review the inputs that we'll use. We'll have 4 different conditions, untreated and treated with afferent A. And then we have our glucocorticoid resistant cells and then our glucocorticoid sensitive cells. So 4 groups, 3 samples in each group. And what we've done is we created an accession list with all the files we want to download from Geo. We're actually going to download the FASTQ files. So these are rather large files that we can pull into the platform and then process. But you can imagine you could pull in your own FASTQ files from your own sources. We also have a sample sheet, which is obviously very important when doing any type of research. We have the different conditions. Each file has been labeled with its actual condition. So we'll use that in our processing as well as BigOmics. And then the output from Genome will be a series of reports that you can review the data. And then, of course, the counts that will, the counts for each gene that can be used in downstream analysis, such as BigOmics. So without further ado, I'll dig into the Genome platform. All right. Hopefully you can see my screen. So this is the Genome login page. And so here we can see all of the different projects that are available to me. And this is how I can organize and store my data, my projects, and my workflows. We can create new projects or launch Jupyter Notebooks for more advanced analyses. But I'm just going to start off by creating a simple project to download the multiple data files. I can select an icon and then create a new project. And so this is a blank project, and we can see we have several tabs here, details page where we can see a summary information about the project itself. This is where we can connect our different data sources. So this is really important. It allows us to connect to either an S3 storage directory that we have available, or it can allow us to upload files from our own computer. And so, that's a nice feature because we can actually upload quite large files into the Genome platform if you have them. So today I'm just gonna do a quick example of uploading the two files that we talked about, that list of accession numbers, as well as the sample sheet. So quickly upload them. And now we can see that they're available within the Genome platform for us to use. Great. Now I'm going to dig into the Genome language and basically the power of building workflows in Genome. So this is really important part of the platform. And so I'm going to start by going to the Add Tools section. And I'm going to explain a little bit what these are. And so I'm typing FastP, and you can see this is a wrapped tool. It's a containerized tool that contains everything you need to do, everything you need to have to run this tool in the cloud. And so it's completely containerized. It's version controlled. It has documentation, and we can add this to our workflows, which I'll show you in a second, where we can actually build complete workflows and process data from FASTQ all the way through to process data. And so you can pull in, you can imagine you can pull different tools together to string them along to build your workflow. We're going to start off actually by bringing in a pre-built workflow. Import. And so we have a workflow package that we've built out to allow you to pull in bulk RNA sequencing data from Geo. So we'll simply add this to our project. It's been added. And I'm going to show you, so the plug and play is kind of a simplified version where you can just connect things and it's a single tile. I'm going to show you the full workflow here. And we can see we have our inputs, our variables, and then we have these tiles that encapsulate different functions or tools. And so in this example, we have our SRA toolkit, which is a common tool used to pull data from the sequencing reads database. And so we can fetch it, and then it'll connect using these wires to different functions downstream. And so through this process, we can build workflows visually without the need to code or worry about an environment. We can just build workflows visually and not have to worry about cloud infrastructure or any of the other things that you have to do when you're actually writing code in a text editor. So what we're going to do is just something very simple. We'll delete this input pipe, and we're going to just pull our accession list to the Canvas workspace and then connect it up. And now we've connected this accession list to the workflow. We can save it. You can see everything's version controlled here. Save the workflow, and then run it. And it'll do it'll basically spit up all the resources in the cloud that it needs to basically pull down this data. And it'll do so in a parallelized process. So it's not waiting to do each one individually. It'll do everything together, spin up all the resources. So every file that it gets, it can just process that file on its own, and so parallelized. So that's a really important feature. It saves a lot of time. Things are done quickly because it can spin up the exact resources it needs for each tool and then shut them down when they're done. No need to keep some sort of instance or cloud computer open in perpetuity to analyze a project. So I could run it here, but it will take some time because it's actually a very large data set. And so I'm going to back out of here and show you one that's already been downloaded. And so just go to our data files. You can see we have data set and then the project number. It's 81 gigabytes, so it's actually quite large. And you can see it contains all of the FASTQ files. We actually have 24 items, 12 samples, paired in reads, so there's 24 files. And it's compressed all of those FASTQ files to conserve space. And now I'm going to walk through setting up a guided workflow. So a guided workflow is a workflow that has been wrapped in a form, and we're basically guiding the user through the questions and the things needed to set up the run. And so it's a much more simplified process. Importantly, we can build guided workflows from a Canvas workflow, or we can actually wrap them in Nextflow around Nextflow workflows. So the platform is compatible with Nextflow, and so people can use that as their backend processing pipeline. And so we can build these guided workflows, which are kind of easy ways to set up processing runs, for their sequencing data. So we're going to run the guided workflow. We have basically three guided workflows. Two for single cell RNA sequencing, and then one for bulk RNA sequencing. And so I'll select that one. It gives it a default name, but we can change it. Change the name if we like, and then we just select our data set. So very easy. The great thing here is that it pairs the reads for you, so you don't have to do the pairing. You can change the sample names if you like, and if there's ones that you don't want to include in your processing, you can just simply delete those. But we'll keep everything. And then here's an important step where we can use the metadata that we know of our samples, our 4 conditions, and label each one of these files. Sometimes it's easy enough to do it manually. You can select control in your experimental groups or change the name to treatment or some other variable. But we're actually going to use that metadata file that we had previously. And that's a nice way of keeping track of all the metadata when you're dealing with a large number of files. And so we can see here, they've all been set to the appropriate group. So next, we can select the reference genome that we'd like. So I'll just use reference gene set genome 37. The count method can vary depending on the application that you're doing. And so we give the user the option to pick between the 2 most popular ones, and we'll just keep it on feature count for today. And then here we have different variables, optional parameters that you can set. These are common parameters that people want to change depending on their analysis. And so this will do static analysis, and I'll show you what that looks like here in a second. But you can set these up, and then it gives you a nice summary to what you'll be running. And so this will spin up again all the resources you need to process these 12 samples, and then I'll share with you the output. I won't launch it here because I've already run it, but I can share with you the output of the guided workflow. And so here I have a completed run. We can view the results. So this is a great way where it's been organized for you. And we can walk through this. We have a few things I want to just point out. We have a pre-trimmed and trimmed alignment reports. So these are the multi-QC reports for the data sets. And I'll just show you what it looks like after trimming. It gives you the full multi-QC report for every file and every sample here. And probably the most interesting to see is, we have a great % alignment. So everything's aligning to something in the human genome, which is what we expect. We have a high number of duplications, which, I think just means that the sample's been over sequenced. You can see in this graph, they've sequenced about 24, 25 million reads. About half of those are duplicate reads. So a little bit over sequenced, but nothing to worry about. The data is still great to use. Another thing that I want to share with you is the differential expression report. And so this is a feature within the genome guided workflows where we can actually output summary results as in the form of a report. This is really nice for routine workflows or things where you need just an organized answer, whether you're doing some sort of test or a standard assay in your lab, and we can build these reports. This one's for bulk RNA sequencing, and you can see at the top, it gives you a nice overview of the report and what it has to offer. And the first plot we see is a PCA plot. And so Real nice here to see that everything's segregated into 4 quadrants, exactly what you would expect with this type of cell line data. So very nice. We have other plots like an MA plot. You can switch between different comparisons here. And so this is a downloadable HTML file. You can email this, you can send it to your friends and colleagues. Obviously a heat map and a volcano plots where you can, a static volcano plots where you can see the results. You also get other information as well, individual files. So if you want to look at, say, a heat map that DEC2 generated, you can do that, and we can view it here. A little more contrast, so you can clearly see, the 4 distinct expression profiles from the top 25 differentially expressed genes. So really nice to see those differences in your data, in any data. And then I think the final thing I'll share with you is that gene counts data. And this is simply the place where you can download the files and you have your counts files, you have your sample sheet, and you can simply download them. It'll give you a little pop-up, telling you how big the download will be in case it's a large amount of data, and confirm it, and you'll download that. And this is what you'll hand off to BigOmics. So thank you for your attention, and I'll pass it over to Axel.
Yes, thank you, Kit. And hello, everyone. I'm Axel Martinelli, and I'm the head of biology at BigOmics Analytics. And basically, with our platform, we can take over from where the Almaden platform stops with its analysis. And in particular, we can take the counts CSV file that Kit showed you before, and then we can start doing some more in-depth tertiary analysis and visualization of the data. So let me show you how our platform works. So this is where you input the data. So this is cloud-based solution, of course, and you can see here we can select the species, and we can see we now support around 120 species. Now the one we are working with is human, so I'm going to select that one. And now here, what I can do is go to the data set that we are analyzing today, and I can select the counts CSV file that was produced by the Almaden platform. And then I can combine it with 2 other outputs, 2 other inputs that I will describe in a moment. So let me first of all load the data into the platform. Now you can see that the platform will provide you with a preview. So this is the read counts table that came from the Almaden platform. And the good thing is, by using the Almaden platform, you already know this is in the correct format for our platform. So it saves you really a lot of optimization that you might otherwise need to do. The samples files is very simple. It just contains the phenotype of your data in a CSV format, which you can easily prepare with Excel. And here you can see the sample IDs. And then we have, for example, glucocorticoid resistant or sensitive phenotypes and treatment. The contrast files is an optional file that you can also generate using Excel. And I'm showing it here to you here because it's one of the optional inputs, but you actually don't need to create that with Excel. We now have a graphics user interface that you can use to generate your pairwise comparisons. So for example, if I go here, I can select from one of the phenotypes. I already created an intersection between phenotype and treatment, but I can also just select glucocorticoid phenotype and treatment, and then you see that the platform generates an intersection between these 2 groups. And then it's just a matter of clicking and dragging. So for example, we can use the resistant untreated as the main group and the sensitive untreated as the control group. And the platform will generate a name here that you can edit. Oops. Well, let's just do something simple. Just stick to this. And then you can add this at the end as a new pairwise comparison just by adding click comparison. So once you have done that, you can go to compute. Now, you can select the name of your data set. You can select a description of the data set, the type of data you're working with. So our platform supports both transcriptomics and proteomics data, as you can see, and it supports different technologies for all these data types. So, for example, we go from mass spec to Olink data. This is all supported by your platform. And then all you have to do is click compute. Now, for this example, this is bulk RNA-seq data, so the default options will work absolutely fine, and you don't have to worry about anything else. But for other data types, we also have additional options that you can choose. So I will maybe speak about these advanced options if there are any questions about it. So once you click compute, it will take between 5 minutes to 45 minutes, depending on the size of your data. And then once the data has been fully uploaded, the platform will take you to the main page. And as you can see, I've been quite busy in the past few years, so I have uploaded quite a few public data sets. I already loaded the data set that we are going to work with today in memories. It's this one. Also, a nice feature that I quickly want to show you here is that through our platform, it is possible to now share the object files that contains the figures and the tables directly with other users. So all you have to do is just select an email address and then share the data set. And even if the users don't have an account, they will be invited to create a free trial account through which they can take a look at the data. Now, you can see that in terms of visualization, the platform is divided into 6 sections. And for the purpose of the presentation today, I'm going to go through those modules that are relevant for the analysis that we are doing. And I'm going to use that both to explain a bit the nature of the data, but also to show you how you can interact with the platform. So the idea, the principal guiding motive behind our platform were twofold. We wanted to make the platform both highly interactive so that you can modify the plots on the go as you work with them. And secondly, same principle as Almaden, make it as user-friendly as possible so you don't need to do any coding. Everything is fairly intuitive and accessible to users. So let's, for example, look at the dimensionality reduction. So as you can see here, the structure of our platform is that we have always on the right-hand side a settings panel that applies parameters to all the plots that you see in a page. And then if you see here on top of each plot, we will have usually 4 icons. And the icons here, the 3 horizontal lines, represents the settings that will be applied to the plot in questions only. So let's go first to the advanced options here and choose a different layout. So we have by default the UMAP layout. It's ideal for single cell data, but we are working with bulk RNA-seq data, so we want a PCA plot. So now we are seeing that the default choice is showing me the clusters. I want actually to see my intersection of phenotype and treatment. So I'm going to select this phenotype. And now you can see exactly the same that you saw with the Almaden platform. You can see that we have 4 discrete clusters representing the different treatment and glucocorticoid phenotype groups. And as I said, our platform is highly interactive. So for example, if we can turn this into a legend that we can add at the bottom, so we don't have the names on top of the samples. And we can also easily turn this into an interactive 3D plot, which you can see here. This is something you can zoom in and out, and we can easily visualize and manipulate. Once you're happy with the visualizations, you can then download the data, and you can download the data in 3 formats. First of all, for your presentations or your papers, you can download publication-ready PNG and PDF files. If you're a more advanced user and you want to have access to the raw data that went into producing the plots, you can download a CSV file with the raw data that allows you to recreate the plots using your own R pipelines, for example, or use this data for further analysis with other approaches you might want to apply. So same principle applies to the heat map analysis. We have two types of heat maps. We have, first of all, a dynamic heat map where you can mouse over, and then we have a more traditional static heat map. And again, here you can see reflected the same type of structure that we saw with the Almaden platform, where we can clearly see differences between the samples, and we can recognize 4 blocks that correspond to the different conditions. Now, we can simplify the visualization of these heat maps going again to the settings menu on the right-hand side. And we can say, well, I really want to emphasize one of the phenotypes. And in this case, I want to emphasize the intersection between phenotype and treatment. So I'm going to select this phenotype. And now you can see that in a couple of seconds, the platform will rearrange the samples based on their treatment and phenotype status. So we can see here that we now have the sensitive samples on the left-hand side and the resistant samples on the right-hand side. And we can see there is this block of genes that distinguishes treated versus untreated, and then there is this block of genes here, for example, that distinguishes resistant versus susceptible. Oh, sorry, the other way around. So now, again, you can interact with the plots. There are different options. I'm not going to really go through all of them right now. But what I want to draw your attention to is that the platform will also automatically generate some gene modules based on gene expression. And you can see here that it has produced 4 gene modules. And in particular, S1, S2, and S3 are interesting because they distinguish the different phenotypes. So now we can go to the functional notation, and this is again something that you can customize. So if we go to the settings here, you will see that the reference set that we are using by default is the hallmark collection. However, if you click on it, you will realize that our platform offers you actually an ample choice of public databases that you can use for your annotation. So for example, we have the Go pathways that most of you will be familiar with, but those are more exotic ones, such as aging-specific or drug-specific ones, transcription factor-specific ones, and so on. So in total, we have more than 50,000 gene sets and pathways that you can use for your functional annotation. And if we check here now, we can see, for example, the S2 and S3 terms that were related with if I'm not wrong, let me just make sure. Yeah, S2 is related to sensitivity versus resistance. So you can see some terms that are also related to tumors, such as cross-signaling pathways, p53 pathways, also UV responses going up. And then we also have S1, which is related to treatment versus untreated. And you can see, obviously, some typical stress responses due to the action of the drug, such as UV responses, also hypoxia. It's a typical signal of stress. So we can already see that there is some information here that makes sense. Something a bit unique about our platform is that we can actually perform clustering analysis at the gene or gene set level rather than at the sample level. And this is maybe something that some of you might not be familiar with, because we are all used to work with sample level analysis. So what we do here is basically order the genes in a plot, in a UMAP plot, based on their expression profile across all the samples. And then we color the genes or the gene sets, as you will see in a moment, based on the level of variation across the samples. So we can immediately identify hotspots of variations, which are interesting from a functional point of view. Now, let me go straight to the gene set analysis, because I really want to show you this, as you will immediately see why this is so informative. And also, very visual, very simple to work with and understand. So here we can see a clear hotspot of variation between untreated and treated samples. So here we have all the treated samples, all the untreated, and you can see these gene sets are upregulated under treatment, downregulated in untreated samples. So we can actually highlight this area on the platform, and we get a list of all the gene sets and pathways that are affected, in this case, upregulated after treatment. Not surprisingly, so remember here we are combining all the 50,000 gene sets. You can also customize this. You can select to focus, say, only on hallmarks or go terms, if you want just to look at pathways rather than, in this case, all the drugs that show similar profiles. But here, not surprisingly, it shows a lot of drugs generating similar profiles. And if you look here, you can see that with heparin itself, the drug that we use in our experiments, is also shown as showing a positive correlation with the data set. So it correctly identified this as a with heparin treated sample. And there is also, of course, a lot of more interesting information that we can then explore further in the other modules of the platform. I'll just quickly show you that within each of the segments, we cover a lot of analysis. So we cover, for example, differential gene expression that you already saw in the Almaden platform. We produce also volcano and MA plots. If you select a specific genes, you can then visualize the gene in isolation in the plots. You get also plots specific to the pairwise comparison you're looking at. So let me maybe switch this to treated versus untreated just to make it a bit easier. And here, for example, we can see, not surprisingly, this shock protein A6 as the topmost upregulated one after treatment with heparin A. And we can also see an overview of all the pairwise comparisons. And you can see every time I'm comparing treated versus untreated, this shock protein A6 go up. When I'm comparing sensitive versus resistance, not much going on. Also, a quick note here that I want to make sure. So again, you can interact, you can choose the FDR values, the log fold changes, but I also want to draw your attention to the star system. And this is because when you run the platform with the default options, it will automatically select 3 different statistical methods to identify the up and down regulated genes. And the platform will not just treat them in isolation, it will combine them, intersect them to provide you with greater confidence in the robustness of your analysis. So this time, when you see a gene that shows 3 stars, indicates that all 3 methods identified it as significantly upregulated. And this is something you can customize. If you deselect 2 of them, you can repeat the analysis just with one of the algorithms. So if you overestimated the strength of your signals, that's not a problem. Find biomarkers is something that I use very often, even if I'm not looking for biomarkers, because it can sometimes provide you with interesting functional information about your data set. And I want to show you this in correlation with the combination of phenotype and treatment in this data set. So this plot generates, this module generates 4 plots. First of all, a decision tree that tells you, okay, you can use these 3 genes to distinguish your different groups. So you can have a shock protein which distinguishes untreated versus treated, as you can see here. And then we can use these 2 genes to distinguish the sensitive versus resistant conditions. And if we then look at the biomarker expression of the top 8 potential biomarkers, you can see the profile of expression across the different conditions. So for example, you can clearly see heat shock protein A6 increases in response to treatment. Also quite interesting, you see this pretty unique profile for DNA JA4, which is another heat shock protein, but has a different function. This is actually a chaperone for exporting proteins on the surface of the cells. And you can see that unlike the others, it's only upregulated in the sensitive samples that are treated with heparin. And you see there is no increase in expression in the resistant samples. And the interesting thing is that a related gene is involved in the transport of glucocorticoid receptors to the surface of the cell. So this is not just a biomarker, but could also underlie the mechanism of glucocorticoid resistance in this particular example. So this is always something that I like to show. And this is just to explain how we do this. We use a combination of different statistical approaches. We use 6 machine learning algorithms as well as 2 other statistical tests. And then, as you can see, this is a sort of guiding principle behind the platform. We combine all these different approaches. In this case, we produce a cumulative variable importance score to show which of the biomarkers were identified by which of the approaches. And in this case, for example, you can see HOC protein A6 being identified by all 8 of the approaches. Of course, we cover gene set enrichment analysis quite exhaustively. We do this by producing this gene set enrichment plot. So this is showing an upregulation. Let's pick again treated versus untreated. So you will see that now we have an upregulation mostly related to different drug signatures. And then if we turn it upside down, we can see the reverse. We can see a downregulation of a specific gene sets. And if you select specific gene sets, you can also see the different components and how they're up or downregulated in relation to the control group that we are using here. So again, if you see here, we have a 3-star system, or in this case, 2-star systems, because we are using just 2 approaches for analysis. And same principle, you can deselect it. You can also select to focus on a specific gene collection. If you think, this is too many entries, I need to trim them down, or I just want to see all mark collection, then it's something you can do. Related to that, we have also pathway analysis based on 2 main collections, Wikipathways, which is open source community-driven alternative to KEGG pathways, and the Reactome pathway collection. And we also have a visual representation of the Go terms. So again, let me go to treated versus untreated. And here you can see a typical Wikipathway plot. In this case, not surprisingly, oxidative stress response. Again, it's typical response to stress induced by the drug. And you can see there is a massive upregulation of several components. And with these plots, you just get a nice visual overview of exactly what components are affected rather than just a list of genes. As you go down the list, you can see all the different aspects that are affected here. We can also do exactly the same at the Reactome level. We can then also look at the Go terms that are up or down regulated, and we produce these interactive gene ontology graphs that you can play with on the plot and arrange in a way that you can then save as an image. Also, what we produce here are different activation matrices that shows you an overview of the most up and down regulated, in this case, Go terms, across all the pairwise comparisons that you're looking at. And here, for example, you can see that all the all the treated versus untreated comparisons show downregulation of this block of Go terms. So you can already see the similarities and difference between them. Another very powerful feature of the platform is being able to compare signatures. And you probably are used to do something, quite classical, which is producing Venn diagrams, looking at different pairwise comparisons within your data set, trying to find a core of activated genes that you can then use for further analysis. Of course, our platform can do that. But we decided, well, why limit ourselves to just looking at the data set in isolation when we have so much data in the public realm and data that we also upload into the platform that we could use for comparative analysis. And this is exactly what we did. We can either compare your data sets with all the other data sets that you uploaded into the platform, or as I'm showing you here, you can also access a collection of more than 6,000 public experiments collected from the GEO database that you can then query across all their pairwise comparisons to find which of these pairwise comparisons gives you the closest match in terms of gene expression profile to the pairwise comparisons we are looking at here, in this case, treated versus untreated. And you can see, not surprisingly, that the first match that I find is from another experiment using bortezomib, which is a drug that is frequently used to treat multiple myeloma, and you can see there is a nice positive correlation. It's also interesting to notice that bortezomib is a proteasome inhibitor, and with efferent A also acts, among other things. It's a natural compound, so it has multiple modes of actions, but one of them is inhibition of the proteasome. So you can see that the platform already identified a good match that can tell you a lot about the nature of the drug. And once you have a data set you're interested in, you can then look at it more in details by going here, selecting a data set from the list of uploaded data sets, and then do a one-to-one pairwise comparisons between the data set, which extends to looking at both these plots, these UMAP plots, but you can change that. Now I'm just going to pick a random data set just to show you that this will change according to the data set that you upload. But it basically allows you to then look more specifically at the individual genes and how they are behaving. So here, for example, you can see that the two plots don't match. So there is not really a strong correlation, which you can also see, for example, at the fold change level. Whereas if I had chosen the bortezomib data set, you would see a nice strong correlation. Finally, under systems biology, we have some features that are more dataset specific. So for example, cell profiling is really suitable for single cell RNA-seq data. WGCNA is something that you can do if you're interested in producing gene network analysis. But for this particular presentation, I'm going to focus on the drug connectivity map, because one of the things that you might be interested in doing in the course of your experiments is twofold. A, you might want to understand what's the mode of action of my novel, extract from this tropical plant that I discovered has anti-tumoral properties. The second thing is, well, I have this condition. Is there in the public database a drug I could repurpose to treat it? And with our platform, you can answer both of these questions, as well as look at individual genes that have been knocked out or overexpressed and see how they can relate, for example, to the activity of your drug. So they might, for example, indicate some other functional explanation for how your drug works. So if I look here at my treated versus untreated sample, I can see a lot of drugs that show positive correlation. Now, this is quite interesting because we produce a meta-analysis here. So we are not looking at a single experiment. We are looking at all the experiments that were done in this case with cucurbitacin. So it's really a meta-analysis, unlike what you get from Enricher. And already you can start seeing, okay, is there some, similar mode of action that I can discover here? But to make your life even easier, because for most of the drugs, we know the mode of action, we can actually combine all these drug analysis in this plot here, and just making sure, yes, I got it right. We can combine all these drugs by their mode of action and then tell you these are the drugs that shows the most positive correlation with mRNA, and these are obviously the most negative ones. And you can see, for example, here, when we look at this mTOR inhibition, that's one of the properties of with mRNA. This is also related to the proteasome inhibition, and you can see, PI3K, obviously mTOR and PI3K are correlated. With mRNA in traditional Ayurvedic medicine is used as an anti-inflammatory molecule, which is captured by its properties as an NF-κB pathway inhibitor. Some papers also describe these properties as a histone deacetylase inhibitor, as a heat shock protein inhibitor. So you can see already that in the top 6 to 7 modes of actions that the platform describes here, we capture a lot of the functionalities of a natural compound. And of course, we are talking about a natural compound. So if you're looking for a specific small molecules, you will find even more precise answers. Now, if we were trying to find, for example, drugs that, on the other hand, could reverse resistance, right? This is the other big question that we want to answer here. Well, can we find a drug that we could use to reverse glucocorticoid resistance? So in this case, we want to do the opposite. We want to look at a drug that has a negative profile. And then we can go through the list of drugs here. You can see a lot of compounds that have, no experimental compound. But one of the interesting one that I found in my study was this one, strofantidine. And you can see strofantidine as a strong inhibitory component against resistance to glucocorticoids. And now, if you check the literature specifically for these drugs, you will find that it has actually been used in a different type of cancer, but closely related, leukemia, to reverse glucocorticoid resistance. So the platform already picked a candidate that has actually been used to reverse glucocorticoid resistance in one type of cancer. Now, there are other functionalities that we can use here. So you can, as I mentioned, you can also look at individual gene knockouts or overexpressions. You can also look at drug sensitivity map, which is something that you can use, for example, to find good partner drugs. And I will stop here with my presentation, leave some time for questions. Thank you.
Thank you so much, Axel and Kit, for the amazing presentation. So we do have some questions, and I want to open up the floor for any questions. If you guys have anything to ask, please put it in the chat box. And if you do ask it anonymously and we don't get to address it live, we will not be able to follow up. So that's just my one recommendation, is if you would like a follow-up, please make sure it's not anonymous. We do have some questions already in the chat box. This first one, Kit, I think you're probably good to handle. What if we have data from different studies? How can we normalize our data in your platform? How can I supervise over the methods employed by the analysis?
Great question, and I'll answer it from a genome perspective, and then pass it to Axel for a BigOmics perspective. But in genome, you can process all of these different studies through a single workflow. Oftentimes different data sets are processed in various ways with different tools. In genome, you could do it all through the same pipeline, so you have confidence that it's all been processed the same way. And then you can use different tools to help you kind of normalize if you need to, or pass on the raw counts to BigOmics. You can, it has access controls within genome. So if you have people in your lab that are also using genome, you can see the workflows that they're building, and you can share those with each other so that you can keep track of who's using which tools and which pipelines, and interact with people's studies. In our platform, you can actually pull multiple data sets into a single study and combine that together.
Well, from the BigOmics point of view, of course, as I showed you before, we have a module dedicated to comparative analysis across data sets. And the one thing I forgot to mention is that you can use this not only to compare transcriptomics data with themselves, you can also use that to compare, say, transcriptomics data with proteomics data. So it's a bit of a, if you wish, a beginner's multi-omics solution.
Thanks. And I think as one more follow-up to that question is, how would you recommend for comparative analysis when you have both RNA-seq and proteomics data?
So the main difference is how you perform the calculations, right? You have to choose slightly different parameters for proteomics and transcriptomics data. There is also most of the times when people come with proteomics data, they use their internal normalization approaches. So with our platform, you can skip the normalization approach and use the one that your provider gave you. And then once all the calculations are done, everything is presented in exactly the same way. That's what allows us to compare directly proteomics with transcriptomics data. So although the calculations are done differently, then the data is summarized in the same way, in this case, at the gene or protein level.
Great. Okay. Another one for you, Axel, and then Kit, maybe to chime in as well. For single cell analysis, does BigOmics generate comparable clustering as Surat and Scampi?
Yeah, I mean, in fact, Surat is one of the approaches that we have integrated in our platform for doing the cell type profiling. So I haven't really shown it because of with this data set, we focused on bulk RNA-seq data, but we use 6 different reference databases for the functional for the cell type profiling. We have, of course, 4 dedicated to immune cells. Those are actually 7, I should have said, and then 2 dedicated to different cell types and 1 for tissue specificity. The only thing I will add when you work with single cell data with Omics Playground is that you have to do some subsampling at the moment. But this is something that in the future you won't need to do.
Great.
Yeah,
Thanks.
and I'll just follow up on that. You can also process your data in Genome, your single cell data sets. And so you can pull in those ScanPy tools or the Seurat tools and analyze them. We actually have some pre-built workflows and guided workflows for that to prepare their data for either analysis and build reports in Genome or exporting them, of course, to BigOmics.
Thanks. For proteomics data, should we do our internal normalization or just induce the raw data in the platform?
You can do either. It depends. I would say it depends. If you have always been working with a certain type of normalizations and you want to keep it consistent, then I would suggest keep using that and just skip the normalization step. If on the other, you're starting from scratch, I would say just use the platform's normalizations as your default.
Okay, this one's for you. Can you analyze public data on Genome?
Yeah, absolutely. We pulled in data today from the gene expression omnibus, but you can pull in data from other data sources as well. This is TCGA.
Okay. Can I upload a custom reference genome?
For genome, absolutely. If you are working with another genome, whether it be human or oxalot or whatever you're working with, you can do that in Genome.
Great. Okay, Axel, this one's for you. Can you pull functional or pathway level insights from BigOmics?
Yes, absolutely. As I showed in the platform, we do both extensive gene set enrichment analysis and pathway analysis. I didn't really show it during my presentations, but you can also upload your own custom gene set collections, or any gene set collections that are not present in the platform can be uploaded separately as another input file.
Okay. And then sort of a follow-up, can you export all of the figures from the Omics Playground for reporting?
Yes, absolutely. You can export them in PDF and PNG format. Also, you can export the raw data, as I showed, the raw data that went into producing the plots as a CSV file. So you can recreate the file using your own internal pipeline if you so prefer.
Great. Okay, one last question for now. And if you do have additional questions, feel free to type them in the chat box. We can follow up over email. But one last for the webinar, is there any kind of integration between different type of omics, specifically transcriptomics or proteome?
From Omics Playground point of view, yes. As you saw, we have a dedicated module for comparing directly proteomics and transcriptomics data. Also, the analysis that you saw that we do at the transcriptomics levels will be performed at the proteomics level as well. You can actually have, in fact, that's another data set that maybe one day I will show, but we have, for example, a match studies on glioblastoma where we can show the strong correlation between proteomics and transcriptomics data, but also the differences.
Great.
Yeah, and I'll say for Genome, it's a very flexible platform. You can bring in tools for different applications. So if you have single cell data with both site-seq, which is protein sequencing, as well as the gene expression sequencing, you can process all of that data within the platform.
I would even suggest, if you're working with single cell data, combining Genome with Omics Playground, it would be the ideal approach, in my opinion.
Yeah, absolutely. Thanks, Axel.
Thank you both so much. There were a few questions that we didn't get to, but we'll follow up over email. So thank you everyone for joining. We will share this recording and it'll be posted tomorrow. So thanks again to everyone who joined. Have a great one.
Thank you, everybody. Thanks, Axel.
Thank you. Thank you.
Bye.